Good afternoon, students. Good afternoon. Hello, all of you can hear me? Good afternoon.
Yes, sir. We can hear you, sir.
All right. So let me discuss. If something you are facing, you can share with me here in the chat. Okay. All right. So today I am going to discuss with you about biotechnology and genetic engineering. Right. I will do the detail of this lesson. Okay. So let me first share my screen with you. So I think you can see the screen, right? Hello. Ziad, Ihab, you can see the screen, right? Hello. Hello. All right. Yes, we can see, right? All right. So I am going to discuss with you in detail about the biotechnology and genetic engineering, right? So today my topic of discussion is to do about the detail of biotechnology and genetic engineering. Okay. So like in this chapter, I will discuss with you biotechnology and genetic engineering. And then we will discuss about why bacteria are useful in biotechnology and genetic engineering. So why like in the and genetic engineering and biotechnology we are using bacteria. Then we will discuss about different processes that is using in biotechnology, like production of antibiotic, production of like making milk lactose free, and fermenter that we are doing for the penicillin production, right? So all this we will discuss in detail. Then we will discuss about outline the genetic engineering using bacterial production of human protein. Like here we will discuss about insulin production by the help of genetic engineering, in which I will discuss about the different rules of different enzymes like restriction enzyme, that enzyme that do cutting on bacteria DNA, right? And then we will discuss about like how we are inserting that into bacteria. All these processes we will discuss it now, right? All right. Now, then we will discuss about the advantage and disadvantage of genetic modified crops such as soya and maize and rice. Now, the first question, like here I am discussing in a such a way, I will also discuss the exam type questions, and I will discuss their answer with you. The first question with us is why bacteria is useful for biotechnology and genetic engineering. You know, like first of all, you should be clear about biotechnology and genetic engineering. Like what mean by genetic engineering? What does it? Genetic engineering is the like the field of biology in which we are modified the genome of an organism, right? In which we are modified the genome is in which we are modifying the DNA of an organism. Or like we can also say genetic engineering is the process in which, for example, this is a DNA with you, this is a DNA of an organism, and you cut this gene from one organism and insert this gene to another organism, and insert this piece of gene to another organism. So definitely you are inserting one gene, like a fragment, like you are inserting a fragment of DNA from one organism to another organism. So definitely if this organism take this DNA, so it means you, okay, let me mute you because Like, it's very nice. Who is writing? Let me show the name of annotator. Is it clear? All right. So now you actually cut a gene and insert to an organism. You got my point? So, like here, if I put the gene into this organism, it means I modify the genetic makeup of this organism. Genetic makeup means actually I modify the genome of this organism. Like now, the genome of this organism is not natural. So this field of biology is called genetic engineering. Now, is it clear? You can hear me, right? Yes. So this field of biology is called what? Genetic engineering. So genetic engineering is okay. Now is it okay? Is it okay now? Hello, it's okay? Yes, it's okay. Very good. All right. So this field of biology is called what? Genetic engineering, in which you are cutting the gene of an organism and inserting that to another organism. So you are modifying the genome of an organism. You are modified the genome of an organism. So this field of biology is called what? Genetic engineering. Cutting up gene and inserting that genes to another organism. So here you modify the genome or nucleic acid of a organism. So that is called genetic engineering. Now, this we use this genetic engineering when we use, like, what is biotechnology? In biotechnology we are producing certain product for the benefit of human being. In biotechnology we are it's the branch of biology in which we produce certain product for the benefit of mankind. So, like, hello, hello, is it clear to all? Hello, hello. So from here it is clear. Yes, from here it is clear. Okay. So biotechnology is the branch of biology in which we use what we are using. The we use the application of genetic engineering mostly. In biotechnology we are taking help of genetic engineering and to produce certain product for the benefit of human being. So here I think it is clear to you now, like, what is biotechnology? That branch of biology in which we make such product for the benefit of mankind. So in biotechnology, we need the help of genetic engineering and some processes, right? Like when we are producing insulin, so we are using the knowledge of genetic engineering to cut the insulin gene and put that in the bacteria. So that is what genetic engineering. Is it clear to all? All right. Now let me move toward another. Like now I will discuss with you different questions. So the first question with me is, all right, the first question with me is, why bacteria is useful for biotechnology and genetic engineering? Like mostly we are using bacteria in biotechnology, right? And genetic engineering, we are using this to produce product because we insert our gene of interest to the bacteria. So the question is, why we are selecting bacterial cell? Why not we are selecting any other plant cell, animal cell, like that? So the first thing is the bacteria cell grow or reproduce very quickly. So if it is reproduced very quickly, if I put my gene of interest to the bacteria, so the product that in which I'm interested, that product will produced by bacteria very quickly, means I will isolate my product in very less time. Second thing, bacteria having no ethical concern, no ethical issue, no objection over bacteria. If I do it with plant cell or animal cell, right, or use any other organism, some people may do objection on that. So no ethical issue, like if I slaughter the animal, so ethically it is not right to do with that. Okay, is it clear? So no ethical concern with bacteria. Second, bacteria good quality is bacteria possess ring of DNA called plasmid. Bacteria having extra chromosomal ring, like this, bacteria contain this extra chromosomal ring. The chromosome, like it's the bacteria, right? Chromosome, while this one is a bacterial, like it is a extra chromosomal ring. And this is very important in genetic engineering, because we can insert our gene of interest to this chromosomal ring very easily, because it contains different recognition sites for the gene of interest in which I can put in this plasmid easily, and then in bacteria it will express. Is it clear? So because of all these, bacteria is very useful for biotechnology and genetic engineering. I think it is clear to all, like no ethical issue. Bacteria grow, like reproduce very quickly. Is it clear? Is it clear? All right. Now, the bacteria contain plasmid on which I can put my gene of interest easily. So that's why bacteria is very useful for biotechnology and genetic engineering. Now, let me move toward another topic, like it's another question. Describe the rule of n. So before going to this question, I will go to this question and then I will come back toward this question. Why bacteria is useful for biotechnology and genetic engineering? So I did to you this already, right? Like why it is useful. Now, how we can produce insulin, right? Like how, for example, we need insulin to produce insulin with the help of genetic engineering. So these are the basic steps through which you can insert your gene of interest to bacteria, and then in that bacteria your gene of interest will grow. Gene of interest mean the gene in which I am interested. Gene of interest mean the gene in which I am interested. Is it clear? So what does gene of interest mean? The gene in which I am interested. So that gene of interest, I can put it in the bacteria. Okay. So here, let's suppose this is a bacteria cell with you. This is bacteria cell with you. And this is bacterial plasmid. And this is bacterial DNA, right? Like one chromosome, right? Of bacteria. Now, what I am doing, the first step in biotechnology is, this is the human DNA. This is human DNA. And now I am going to, okay? So now what I am going to insert my gene of interest, that is insulin. So now I am going to insert my gene of interest to bacteria, in the end bacteria that gene of interest will express. So here my gene of interest here is what? My gene of interest here is this one. This here, my gene of interest is what? My gene of interest here is this gene. This is a sequence of DNA. This gene code for what? This gene code for insulin. So first of all, I will isolate the DNA of human. Then I will treat that DNA with specific enzymes. and that specific enzyme will cut this DNA, and that specific enzyme will cut this DNA. Okay, is it clear? So that specific enzyme will cut this DNA. Is it clear? Now, after that, when I treat this DNA with restriction enzyme, right? The name of that enzyme is called what? Restriction enzyme. And restriction enzyme are actually molecular scissor. Okay, all right. So let me wait. I will share my screen once again with you. Okay. So I will now. Now, is it okay for all? Let me stop sharing the screen and I will share it again so that if the problem solved. Now, is it okay for all? Just take it now. Is it okay? Hope it is okay for all now. Okay. Now, so what I will do, I will actually cut this gene of interest by treating with restriction enzyme. Let me repeat again because let me repeat again. First of all, I isolate the DNA of human, right? First of all, I isolate the DNA of human, and after that I treat that DNA with restriction enzyme. Restriction enzyme are special enzyme that will cut my gene of interest. So here I cut my gene of interest, and that my gene of interest is human insulin gene. So I cut my gene of interest, that is human insulin gene, and on the same time I also isolated the plasmid. At the same time I also isolated plasmid from bacteria, right? So this is the extra chromosomal ring that I removed from this bacteria. See, this bacteria, from this bacteria I removed the plasmid. Okay, now when I cut this, when I remove this plasmid, I also treat this plasmid. So is it clear? So what I did, I cut this. So when I cut this plasmid with restriction enzyme, and also I cut a gene of interest. Now what I did, I mixed this plasmid plus gene of interest. So when I mix this plasmid in this gene of interest, they will combine with each other by special enzyme that I put already in that solution. That enzyme is called ligase enzyme. They will make a bond between this gene of interest and plasmid. So now if you see here, my plasmid have right now, my plasmid have the gene that my plasmid that I isolated from bacteria that having my gene of interest now. So plasmid DNA with human gene is placed. Now this DNA that I make it by inserting my gene of interest to a plasmid. So this is called recombinant DNA. This is now artificial DNA. So this artificial DNA, then I will insert to bacteria. So there are different methods of inserting artificial DNA or this recombinant DNA to bacteria. So one of the method is I give certain electric shock to bacteria colonies so that their membrane permeability increase, and they will take this plasmid recombinant DNA. Another method is micro injection technique. Through that method, I can insert my this recombinant DNA to bacteria. Now, if you see here, this bacteria contain my gene of interest now. Okay, so this bacteria contain what? My gene of interest now. Now, if this bacteria contain my gene of interest now, so it means the nucleic acid. So, to all of you, the voice is not clear. The problem with, all right. The voice is not clear. So let me stop all the screen sharing and I will start it from the beginning. Okay. Now, what I did, I put my gene of interest in this bacteria. All right. Now, this bacteria contain what now? This contain my gene of interest. So it means now I change the genome of bacteria and it contain my own gene of interest because I put my own gene of interest in the bacteria. Now, this bacteria is called genetic modified bacteria. Now, it is GM bacteria, genetic modified bacteria. Why this is called genetic modified bacteria? Because it contain my gene of interest. Now, what I will do after that, I will grow this bacteria on a media. Media is actually bacterial culture. Media is actually the place where I will provide nutrient to the bacteria and that bacteria will start growing. It will start replicating. And when it starts replicating, it will make copies just like himself, like new copies they will start producing. And when it is making new copies, so definitely if bacteria is replicating, it means they are producing new copy of their own kind. So at that time, my gene of interest, that is insulin, that will also start replicating, and every bacteria there will be insulin. If every bacteria have insulin, it means in every bacteria there will be production of insulin. So my gene of interest insulin will express in that. And by different industrial processes, I will isolate this insulin from that culture. Later on, when bacteria colony grew, many bacteria are produced all with human insulin gene, and these bacteria all will produce human insulin. So this is genetic engineering, right? So the first thing I told you, these are the four important stage, like selection of, like a selection of character. Selection of characteristic means like in which character here you are interested. Here I'm interested in insulin gene. Here I am, here I am interested in what? Here I am interested in insulin gene. So I will isolate the insulin gene by treating it with restriction enzyme, and then the bacteria will start replicating. When bacteria start replicating, look, first I isolate my gene of interest, then I ligate with plasmid, and then I put that plasmid in bacteria, right? Insert that plasmid in bacteria. So in the plasmid, that gene of interest or that bacteria will start like they will start replication. When the bacteria is replicating, so my gene of interest will also replicate with that. Okay, so hope this is clear to all. Anyone question they can ask in the chat. Anyone have question, please study the topic, they can ask here. Anyone have question, they can ask. Hello, anyone have question, they can ask. Yes, please ask it here in the chat, please. Anyone have question regarding this topic, they can ask here. Otherwise, I will ask. What is restriction enzyme? Please. It's a very general question. Yes, tell me in the chat, no problem. Or you want to tell me here, it's okay. Very good, very good. Enzyme that cut the specified gene. Very nice. That cut our gene of interest. Very good. And that also cut plasmid. Very nice. So remember, the gene, the restriction enzyme through which I cut the gene of interest of that human DNA, and the same restriction enzyme I will cut plasmid. Okay, remember, it is very important point. Is it clear? Those, like for example, here I cut this gene of interest by one of restriction enzyme. For example, that in restriction enzyme is BAM-H1. That plasmid I will also cut with BAM-H1 so that it produce same sticky end. So that it produce same sticky end. Is it clear? So that it produce same sticky end, so that both are complementary to each other, and so that my gene of interest ligate with plasmid easily. One of the question here is, I teacher about the ligase enzyme, I did not understand. All right, so let me tell you about the ligase enzyme. You know, when you cut this gene of interest, right, and you mix it with the plasmid, so now both are a separate fragment. So I put ligase enzyme here so that it make bond between this gene of interest and this fragment of plasmid. Like they are making phosphodiester linkage. For example, like this is your plasmid, right? And this is your gene of interest. This is your gene of interest. Now both are separate fragment, right? So the bond that form between these two is called phosphodiester linkage, right? Means two nucleotides are joined together. So the work of ligase enzyme is to make phosphodiester linkage between gene of interest in our plasmid, right? Simple way I will tell you, it act is a glue that combine the gene of interest with the plasmid. Is it clear now? If it's clear, give me response. Is it clear? All right, another question is, yes, it's clear. One question is, okay, what is, all right, now your question is, what is plasmid? So it's very easy. You know, this part of bacteria is called, like, some bacteria, are mostly bacteria, have extra chromosomal ring, like this ring, this one. So this is called plasmid. It is DNA. It is DNA, but not the part of actual chromosome. Like not the part of, like this is bacteria, DNA of bacteria. This is actual DNA of bacteria. But with that, there is another fragment that is called plasmid. It is in the form of ring. And mostly a question here some student can ask, why the bacteria having plasmid? So the answer of that is that, you know, this plasmid contain antibiotic resistance gene. So by that gene, bacteria show resistance to different antibiotics to protect themselves from antibiotics. Is it clear? But molecular biologists take advantage of that, and they isolate that plasmid, and they put our gene of interest in that antibiotic resistant gene places and make their own recombinant DNA. So simply I will tell you that plasmid is extra chromosomal ring. Extra chromosomal ring means it is not the part of actual part of chromosome of DNA, not the part of this, but it is extra ring that is present in bacteria. And we are using that in molecular biology. Is it clear to all? If it's clear, the student who asked question, tell me okay, please. All right, so one question, I should give the answer of this question, right? What is genetic engineering? Anas is asking this question, so definitely I must give the reply of this question. Anas, actually, genetic engineering is the cutting of gene from one organism and transfer that to another organism. Simply change the genome of an organism is called genetic engineering. A very simple way I will tell you again: cutting up genes and ligating up gene. Like here, what we did, we cut the gene of interest from the human DNA and then we ligate that with plasmid. So this is genetic engineering, and then we put it in the bacteria. So I changed the genome of bacteria. So this is genetic engineering, right? Anyone other have question, they can ask, please, regarding the topic, please. Nabil, clear? Very good. Yeah. Actually, here one thing I will just tell you once again. Here I will tell you one thing more. Some student may come in their mind. So I'm trying to also make you clear about this point. Sometimes they also discuss this point. So let me clear you about this point. Yeah, about this point, let's see. Like in the lecture I told you about the sticky end. So I told you that on both, like if I am extracting my gene of interest by one restriction enzyme, I should also treat my plasmid with the same restriction enzyme. Why? So that it make the same sticky end. Sticky end is actually this single strand DNA sit. Look at here. Here the gene of interest is already isolated. This is completely gene of interest. They isolated. But here with this gene of interest, here is a single strand DNA. Like this is single strand, right? And this is also single strand, right? So these are called sticky end. So if you treat your plasmid with same restriction enzyme, so this sticky end will be complementary to this one. So this, if you take this gene of interest and you insert it, so this site will make bond with this one, and this side will make bond with this one. So for example, here we have T, here we have, for example, A, right? So if here is A, so definitely A will make which bond? Hydrogen bond with T. So here I told you, for example, here is A, this place, just look, this place. So here this place supposed to have what? T. So that A will make bond with T, and like that other will make, G will form triple hydrogen bond with C. Like guanine will form triple hydrogen bond with cytosine. Yes, complementary. Yeah, yeah, there should be complementary. Complementary means A will form bond with T and G will form bond with C, and T will form bond, double hydrogen bond with A, right? And what? C will form triple hydrogen bond with C will form with G. So like this, they will the gene of interest will insert. So this is about the sticky end. Remember, if you see the sticky end, so it will be like this. Okay. So these are the steps. If you want to review it, you can review it. Okay. If anyone have question on the screen, this is the explanation. I did to you, but if you want to do like I discussed with you already right now. So if you want to review, so you review the theory, and anyone have question, I can also take the question now regarding this thing. All right. Okay. So it's clear, right? Actually, your question is what are the types of restriction enzyme? So I will not go to that in detail because it is not the part of syllabus. But by the way, I will tell you if you want to ask a question. Like first thing is there is two basic type of restriction enzyme. One is endonuclease, another is exonuclease. Are you getting me? One is endonuclease enzyme, endonuclease enzyme. One is endonuclease, another is exonuclease, right? So endonuclease, for example, you having this DNA, right? And you want to cut the gene of interest from the middle. So endonuclease will cut the gene of interest from the middle. If you want to cut a gene of interest from the end, so at that time you have to use the exonuclease enzyme. Is it clear? If you want to cut at the middle of a DNA, endonuclease. If you want to cut at the inch, you have to use exonuclease enzyme, right? And there is different type, like there is different names of these restriction enzyme, like BAMH1, EcoR1, different types. It's given the name from the organism on which it is isolated. Now, is it clear? If it's clear, the question, give me response. Otherwise, I will do more detail of it. Yes. Student who asked the question, yes, clear, right? Very good. I think clear to all, right? Now, this was the, yes, all right. So this is about the insulin production. Now let me move toward another questions. Okay, so this is about we use, the question is the rule of anaerobic respiration in yeast during production of ethanol for biofuel and bread making, right? So the first thing is we are using yeast in bread making. Why we are using that? Like anaerobic respiration is the process like in which the breakdown of glucose happen and the absence of oxygen, and yeast, the product of that should be carbon, like for in which the product in which we are interested, that will give us carbon dioxide. Here in the bread production, we are like it will give ethanol, but we are carbon dioxide plus ethanol it will give us. But I'm interested here in which, and the production of carbon dioxide. So my question, like the question here that asked from us is what are the steps? So definitely, if first of all I have to take yeast plus the nutrient, like nutrient mean glucose I should provide to the yeast, right? And along with that I will provide them like specific environment, like no, first is like anaerobic environment, warm condition, so that their enzyme work better, right? So they will start the process of fermentation. Fermentation is anaerobic respiration, right? When I mix nutrient like dough with that yeast, water, right? Water also play important role in the activation of enzymes. So when the mixture of both I make it, so what will happen, the yeast will start fermentation, and that will produce carbon dioxide plus ethanol. Carbon dioxide, when it is evaporating, it will make our bread spongy. The dough will become like they will make it soft and spongy, right? So the bread is baked. When the bread is baked, killing the killing the yeast and evaporating the alcohol, right? And holes are left in the mixture where the carbon dioxide gas expanded. Like most of the question coming, like they asking that why we are using yeast in the bread production, so to make the dough spongy. That's why we are using that. Okay. And like same procedure also for that we how we are doing it for the biofuel production. So here, like we mix the barley grain with warm water to germinate it. When it germinate, right? So inside the seed there is starch, so that will convert into glucose, and then we mix that with then we mix that with yeast. Okay, so definitely the yeast enzyme will convert that glucose to alcohol, and when it converts into alcohol, so that alcohol we can use it is for the biofuel product. Like in biofuel we can use that. Okay. Okay, all right. Who's a question? Yes, I will send you this document, don't worry. Okay, if you're having any question regarding the topic, you can ask. Okay. So this is the general mechanism how the anaerobic respiration is happening and why we are using yeast. Okay. So now let's me move toward the questions. This topic I will not discuss it now. So the first question is, state two features of bacteria that make them useful in biotechnology and genetic engineering. So like simple DNA and their ability to make complex molecule. They're like the DNA is simple, but still having ability to make different complex molecule like insulin and whatever we are inserting, they can make it simple. Like another thing, if we can mention it here, they can like replicate at they can reproduce very quickly, so we can isolate a product in which I'm interesting in very less time. Another is presence of extra DNA plasmid. Extra DNA plasmid actually like extra chromosomal ring. In that ring, we can put our gene of interest easily. So another quality of it is like this. Yes, very good, very nice. It doesn't take play space, like it's need very less space, even in a like in a fermenter, like in a very less place, too much like too much replica or too much copies of this bacteria. We can do it in a very less time and very less place, right? So very good, very nice answer. Describe how yeast can be used to make biofuel. So I told you yeast can respire. Yes, reproduce very fast, very good, right? So yeast can respire anaerobically to produce ethanol and carbon dioxide. The glucose can come from the sugar containing waste substance from food manufacture or from the processing of maize grain, like maize grain, barley grain, which you do processes of that, and that glucose then you expose that to yeast, so they will produce ethanol, right? So this like the answer of this question is, describe how the yeast can be used to make biofuel. Yes, very nice. Their genetic information is universal. Very nice, like adenosine, like adenine, thymine, guanine, cytosine, like all of them. It is universal, right? Every organism is like this, right? So that's why. That's why. Is it clear? Very nice. It's also very nice answer. Like their genetic information is universal. Okay, so this question is name the group of molecule to which pectinase belongs. So I will discuss to you a next lecture about the pectinase, right? Okay, they are asking to which like group it belongs. So definitely it is very easy. It belongs to enzyme group. It belongs to enzyme group. Pectinase is okay. So pectinase is enzyme. Okay. Now, another question we have, describe the use of pectinase in fruit juice production. I will do it also in next lecture. Define the term genetic engineering, right? So, okay, one of the student asking question like, what is that? Let me check. Why not the advantage? What's the advantage of biofuel? Is the first thing is like it's like not much expensive. Second, pollution problem will be not more, because of that the people are now producing biofuel. Okay, for the, yeah, for the clarification of fruit juice, yes, we are using that. The fruit juice we are using to mix that with, like we are using, like, you know, when we make the fruit juice, right, that is cloudly because two cells and the plant, they are the, between them there is pectin, and pectin like stabilize both the cell, they join the both cell with each other, right? So if we mix that with the pectinase enzyme, so the pectinase will digest that pectin and will make the juice clear. So that's why we are using it. Okay, let me move because you are interested in this topic, so I will go to that again, right? So they work by breaking down pectin, the jelly-like substance that stick plant silva to each other. The enzyme can also be used to clarify the fruit juice and wine. During the breakdown process, a number of different polysaccharide release, which make the juice like pectin, okay, which make the juice cloudy. But pectinase enzyme will convert that pectin into, that will digest it, and the juice will become clear, and the extract will like become more, right? Another point we have defined the term genetic engineering. So genetic engineering is the changing the genetic material of an organism by removing and changing or inserting individual gene. So this is the answer of the question one of the student asked. Okay, so another question we have, state 3 example of genetic engineering. So the one example I told you already, that we can produce a human insulin by genetic modified bacteria. Second, we can also make genetic modified crops. Certain crops we can make it like so that it produce, we insert our gene of interest in that, and they will produce the product in which I am interested. Like with like vitamin A enriched crops, rice, they already produce, they put the specific gene, and that gene are producing vitamin A, so they produce vitamin A enriched like enriched rice, so they are producing vitamin A. So we can do it like genetic modified crops. We can produce pest resistance crops like that show resistance to feast. Sorry, who is the question? Okay, so these are the examples of genetic engineering. Inshallah, in next class, I will go in detail also in the in the topic of fermenter. Here, name 2 industries that use enzyme and making food. Describe the role of enzyme in these processes. So the first is wine production. Wine production, okay, pectinase is used to clarify wine and industry. Another one is dairy product, and dairy industry, we are using lactase enzyme, right? And that what they do, they make the milk lactose free, right? Some people having the problem of lactose digestion, that are called lactose intolerant. So we immobilize the lactase enzyme and specific beet, and then pass that milk through that beets to that immobilized enzyme, right? And right? And when we are passing our milk to that, like for example, you have special, like this is a vessel or this is syringe, right, in which you put that bead, right? So that bead having what like all that beads we make it, okay, and that bead we put enzyme, immobilized enzyme lactase, and when we are passing our milk through that, so if our milk contain lactase, so that if our milk contain lactose, so that will be that lactase enzyme will break lactose, okay, and it will convert into glucose. Okay, so by this we are making milk lactose free. Okay, so this is enough for today. Now you have 5-6 minutes. Anyone have question? Anyone want any discussion? I can. Yes, they can raise your hand, or I can ask in the chat. Both options are allowed. Okay, both options are allowed. If you want to ask questions, you can write about all the lesson about what is genetic engineering, right? What is the importance of bacteria in genetic engineering? What is the how we are produce, like how we, by which process we are producing insulin. So if you have no question, then I will ask. Yes, please, you can ask question. Ziad Ihab, yes, please. Yes, okay. Another question. I will ask question if you have no question. Okay, my question is, take the question. You are, okay. Why we are producing sticky end? First question is that. Sticky end and genetic engineering. My first question is, why we are producing sticky end and genetic engineering? Okay, another, please give me answer of this. Okay, another question is, what is the difference between, I'm checking your answer all, what is the difference between endonuclease and exonuclease? Take this answer question. Another question is, what is the difference between biotechnology and genetic engineering? Okay, what is the difference between biotechnology and genetic engineering? What is the difference between endonuclease and exonuclease? Okay, what is pectin? Another question is, what is pectin? Very nice. The one question, the one answer I that I Get it now? Exo is for cutting from outside. It's the in. Very good. Very good. Please. Very nice answer. Okay, another question is, please, from this student. What is plasmid? Give me the answer quickly. What is plasmid? To whom I sent, please. Give me the answer. Biotechnology, using information for bio to make good thing for, very good. And genetic engineering, cutting gene and then inserting gene from one organism to and one from one organism and to another organism. Very nice. Very nice. Actually, we are using genetic engineering and biotechnology. Very good. You also give me answer. Very nice. Circular DNA inside the bacteria. Very good. Why bacteria have plasmid? Another question from whom I am asking. Just wait, to whom I will do it. Okay. Why plasmid? Why bacteria have plasmid? Yes, please. To whom I send, please. Why bacteria have plasmid? One of the student, I got the answer. Yes, please. Yes, bacteria have plasmid, okay, for antibiotic resistance gene. Very good, very good. I think it's clear to all, right? So do your work, that work I'm posting to you on the Google Classroom. You have to do that and you have to solve that, okay? And don't worry, I will post after the next lecture. I will also post you this notes, okay? Very good. Basically manipulation of gene. Very nice word you use it, Hashim Dardis. It is very appreciated. I appreciate your word. Manipulation of. Okay, biotechnology is the, but remember, it is not compulsory that in every step we will use genetic engineering and biotechnology. Remember this point, right? Like you are using the yeast. So in there, I mean, it's not compulsory that we will use genetic engineering in every process of biotechnology. For example, you are using yeast and fermentation. Here you are not using genetic engineering, but sometime in biotechnology, we need the help of genetic engineering to produce beneficial product. It should be clear to all, okay? I hope it's the lecture is clear to all. Have a nice day. I will end the meeting now. Okay. Okay. Thank you so much. Have a nice day.